Hello and welcome everyone. My name is Shirsha and today we are going to be discussing about a common lab technique, Southern hybridization, also referred to as Southern blotting. So we have already talked about Western blotting in detail. So we know what essentially we mean when we say the term blotting. So we are going to talk about a variation or a different kind of blotting called Southern hybridization or Southern blotting. So let us discuss briefly about the history of Southern hybridization and its underlying principle. This technique was developed by E.M. Southern in the year 1975 and hence the name. It is named after the inventor of this technique and hence Southern hybridization. And chronologically speaking, Western hybridization actually came about later than Southern hybridization. So because there is already a Southern and then there is another kind of hybridization called Northern hybridization. So the method of detecting or of immobilizing proteins on a blot or on a membrane and detecting it was referred to as Western hybridization. What we don't have is an Eastern hybridization. So someone might come up with something new and in the future we might even have an Eastern hybridization. So a Southern blot is a laboratory method which is used to detect specific DNA molecules from among many other DNA molecules. So if we are to compare Western blotting and Southern blotting side by side, in Western blotting you detect a specific protein molecule from a mixture of different protein molecules, whereas in Southern blotting you detect a specific DNA molecule from a mixture of many different DNA molecules. Now, the DNA that is being detected can either be a single gene or it can even be part of a larger piece of DNA. So, the key to this method is hybridization. Now, what exactly do we mean by hybridization? Nucleic acid hybridization is the formation of a duplex between two complementary sequences. So as we know, nucleic acid sequences are actually complementary in nature and that is what allows them to anneal to each other. So hybridization is the result of this duplex formation between two complementary sequences. So the double-stranded DNA molecule that is formed is actually referred to as a duplex because of the presence of the two strands of DNA. Intermolecular hybridization occurs between two polynucleotide chains which have complementary bases. It can occur between like two strands of DNA. It can occur between one strand of DNA and one strand of RNA. It can also occur between two strands of RNAs. Annealing is another term that is used to describe the hybridization of two complementary molecules. So intermolecular meaning hybridization occurring between two different molecules of polynucleotide chains. Hybridization can also be intramolecular, that is, it can also occur within the same polynucleotide chain provided that complementary regions exist within. So a little bit about hybridization. Heating of the DNA molecule leads to its denaturation. So there's a DNA molecule, or rather two strands of the DNA molecules are held together by non-covalent interactions, and that specifically or mainly hydrogen bonding. And heating leads to the disruption of this non-covalent interaction, holding the two strands of DNA together, resulting in separation of the two strands. And this is referred to as denaturation. So this causes the DNA double helix to open up. Once it is open, renaturation can either occur between the two complementary strands of DNA derived from the same molecule, or it can occur between two complementary strands of DNA derived from different DNA molecules. So in this case, or in the second case, what we end up with would be a hybrid DNA molecule, and hence the term hybridization. So you have DNA from species A, you heat it denatures and opens up. Similarly, DNA from species B, heat it, denatures and opens up. And given and provided that this strand and this strand are complementary to each other, they can anneal together to give rise to a hybrid DNA molecule. So in the context of southern blotting, hybridization involves forming a double-stranded DNA molecule between a single-stranded DNA probe, which would be labeled, and a single-stranded target DNA. So again, if we are to draw comparisons with Western blotting, so in Western blotting, the protein that you want to visualize would be immobilized on the membrane, which can be either nitrocellulose or PVDA. Whereas in case of southern hybridization, the target DNA would be immobilized on the nitrocellulose membrane. Keep in mind that this target DNA has to be single-stranded in nature. Otherwise, your probe cannot go and anneal to it and therefore bind to it. Whereas in Western blotting, you detect or you probe your protein of interest with an antibody specific to that protein, and your antibody is conjugated to a detector, which can be either a fluorophore or an enzyme. Similarly, in case of southern blotting, what you use for probing the blot, that is for identifying your target DNA, is actually another single-stranded DNA piece. Now this probe, which is also a single-stranded DNA molecule, is complementary to the specific target DNA that you want to detect, and this probe is conjugated to the detector, which can either be a fluorophore or it can be an enzyme. Now the reactions are highly specific. The probes will only bind to targets with a complementary sequence. Okay. The probe can find one molecule of target in a mixture of millions of related but non-complementary molecules, and that is how specific this reaction is. Very similar to the specificity exhibited by an antibody for its specific target protein. So probes used in southern blotting. A probe is a short fragment of nucleic acid that, number one, can be labeled with a marker to allow identification as well as quantitation. Number two, will hybridize to another nucleic acid on the basis of base complementarity. So types of labels that are commonly used include radioactive labels. However, radioactive labels are not so common these days owing to the hazards of radioactivity and the hazards or the precautions that one needs to take when dealing with radioactivity, and also because it is dangerous for health in general. So radioactive labels are avoided. But in case one is going for radioactive labels, or previously when radioactive labels were the only available option, so some of the commonly used radioactive isotopes would include P32, S35, C14 and H3. The other type of label commonly used is fluorescent labels, and a classic example of using fluorescent, or classic application rather I would say, of using fluorescently labeled probes is in case of fish, which is fluorescent in situ hybridization. We will briefly, very briefly talk about fish towards the end of this lecture. Or your label can also be biotinylated, and in fact these days for performing southern hybridization the most common label of choice is biotinylated labels. So essentially what happens is that one of the nucleotides that is used in synthesizing the probe, that is either one of the 4 ATGC, are biotinylated, and then this biotin would be used for further labeling and detection. So a little bit about how probes are actually generated. we will be talking about how radioactive or radio-labeled probes were generated, because this is how it all started. But keep in mind that these days radioactive labeling is not so common. It is really uncommon. So the DNA probe is labeled by the random hexamer labeling process. So what does this actually mean? The template DNA is denatured first by boiling. So in order to synthesize a probe to which will be capable of identifying and detecting a specific DNA sequence, you need to have the target DNA sequence to begin with. And because you will be using this target DNA sequence to generate and synthesize the specific probe. So the template DNA is first denatured by boiling it. A mixture of hexamers, that is 6 nucleotides containing all possible sequences, is added and allowed to anneal. So once you denature your template DNA, you add a mixture of hexamers. What are hexamers? Hexamers are nothing but short polynucleotide chains which are only 6 nucleotides long, and this mixture contains of all possible combinations. So this is added and allowed to anneal. Because this is a blind approach and you don't really know exactly which hexamer will go and anneal with your template DNA, but because all possible combinations are present in the hexamer mixture, some of them would definitely go and anneal to your template DNA. Next, DNA polymerase is added along with radioactive nucleotides. So of the 4 nucleotides A, T, G, C, any one of them is taken as in the radioactive form, and for the rest 3 normal non-radioactive nucleotides are taken and DNA polymerase is added. So what DNA polymerase will do is that as the hexamers will go and anneal to their complementary sequence, the DNA polymerase will now extend the hexamer. It will add nucleotides to the hexamer based on the information on the complementary template DNA strand. And since one of the 4 nucleotides in the mixture is radiolabeled, so your probe would end up being radiolabeled. So these days, because biotin is one of the most commonly used labels in place of radioactive labels, one of the nucleotides in this mixture is actually biotinylated. So the mixture is then boiled to separate the strands and is ready for hybridization. Radioactive probes enable autoradiographic detection. So step number 4 actually refers to boiling the mixture after polymerization has been allowed to take place. So you allow polymerization to take place, you once again boil the mixture, and this boiling will denature the complementary DNA strands, so they can now separate out and you can isolate your probe from the mixture. Okay, so these radioactive probes allow for autoradiographic detection. So you basically have your double-stranded DNA, you denature it. Okay, so heating it will separate the 2 strands. You add the random hexanucleotide mixture to it, and because they comprise of all possible combinations of nucleotides, they will anneal somewhere or the other. You add the Klenau fragment, which is your DNA polymerase, and you add your normal DNTPase, and in this case you have this UTP or DUTP, which is radiolabeled. And as DNA polymerase extends this, you will have wherever DUTP is to be, or wherever uracil is to be incorporated, you will have a radioactive nucleotide incorporated. Okay, so this strand, which is produced as a result of the extension of the hexamer, which contains a radiolabeled nucleotide incorporated in it, would actually form the probe. So you do repeated PCR cycles because that will help in amplifying this specific region where the probes have where the hexamers have bound. Or to put it more simply, you subject it to repeated PCR cycles so that you can increase the number of copies of the probe that are being synthesized. So now you will have more and more probe. Okay. So basically after the first PCR cycle, when you go for the second PCR cycle, this extended strand can now also act as a template, and it can allow the synthesis of another complementary strand to it. Okay. So if you want to find out further about probe designing and how probes are generated, and if you're interested in it and if it's something that you plan on doing, then I would suggest that this is actually a very good protocol. You can go through it and it will help clarify any doubts that you might have. So now we will briefly discuss a protocol for Southern blotting and what are the key steps involved in it. So first, restriction endonucleases are used to cut the high molecular weight DNA strands into smaller fragments, because obviously if you just isolate entire genomic DNA and load it onto an agarose gel, it will not even enter the gel, let alone resolve. So first you have to digest your DNA to break it down into small fragments, which can then pass through the agarose gel and would be resolved on the basis of their molecular weight or their size after the gel is run. So the DNA fragments are then electrophoresed on an agarose gel to separate them by size. So similar to what we do when performing a Western blot, you have your protein mixture, you run it on an SDS page to separate the proteins. So similarly over here you have a mixture of DNA fragments, you run it on an agarose gel to separate them on the basis of their size. So if some of the DNA fragments are larger than 15 kb, then prior to blotting, the gel may be treated with an acid such as dilute HCl. So why do we do this? This will depurinate the DNA fragments, breaking it into smaller pieces, therefore allowing more efficient transfer from the gel to the membrane. Because you have already digested the DNA with a restriction enzyme, and it can be quite possible that the location of the restriction enzyme cut sites are so placed that you end up with large DNA fragments. Under such situations, to allow efficient transfer from the gel to the membrane, the gel after running is treated with dilute HCl, which will depurinate the DNA fragments and therefore break them into smaller pieces. Now, if alkaline transfer methods are used, then the DNA gel is placed into an alkaline solution, typically containing sodium hydroxide, to denature the double-stranded DNA, because you require your immobilized DNA on the blot to be single-stranded in nature so that the probe can anneal with it. So for alkaline transfer methods, this alkaline solution helps in denaturation of the double-stranded DNA. Now, the denaturation in an alkaline environment may improve binding of the negatively charged thymine residues of DNA to a positively charged amino groups of membrane, separating it into single DNA strands for later hybridization to the probe, and destroys any residual RNA that may still be present in the DNA. Please ignore this line, see below. It's a typographical error. So essentially, the membrane that is used for Southern blotting is a nitrocellulose membrane. Nitrocellulose membranes are positively charged, and this helps them to interact with the negatively charged DNA molecule. Also, if you're performing alkaline transfer methods, then these conditions help in removing any residual RNA that might be present in the DNA that you have isolated. So this will ensure that the RNA which might otherwise interfere with detection of your DNA fragment of interest is removed. Okay, and this alkaline environment, because it denatures the double-stranded DNA, it improves binding of the DNA to the membrane or the blot. Now, the choice of alkaline over neutral transfer methods, however, is often empirical and may result in equivalent results. So it is not really necessary that you have to go for alkaline transfer. You can go for neutral transfer methods as well. However, this again you have to decide based on your specific sample, that which of these two transfer methods is best suited for your sample. So a sheet of nitrocellulose or alternatively nylon membrane is placed either on top or below the gel, depending on the direction of the transfer. So again, you have to keep in mind that your DNA molecules are negatively charged, so they will migrate towards the positively charged electrode, that is towards the cathode. So your placement of the membrane with respect to the gel should be such that the membrane is on the the membrane is towards the cathode with respect to the gel. So it can be either on top of the gel or below, depending on how the electrodes are arranged in your transfer apparatus. So pressure is applied evenly to the gel to ensure that there is even contact between the gel and the membrane, and to ensure that there are no bubbles in between the membrane and the gel. If any bubbles are entrapped, that will interfere with the process of transfer. So even pressure is applied to remove any air bubbles that might be entrapped in between, and typically a stack of paper towels as well as a heavy weight is placed on top of the membrane and gel to ensure the uniform pressure. So buffer transfer by capillary action from a region of high water potential to a region of low water potential, that is usually you have the filter paper and paper tissue stacks. So some of them are soaked and the other, that is the ones placed towards the well, I will make it will become clearer when we see the schematic. So let us go with it in such a way that the ones which are furthest away, they are dry and they are not soaked, whereas the ones which are closest to the membrane are soaked. So as the capillary action or as capillary pressure makes the water molecules migrate from the soaked towards the dry filter papers, this capillary action will along with it move the DNA molecules as well from the gel onto the membrane. So essentially the transfer actually occurs under the action of capillary movement aided by electric current which is applied. So ion exchange interactions will bind the DNA to the membrane due to the negative charge of the DNA and the positive charge of the membrane. The membrane is then baked in vacuum or a regular oven at 80 degrees centigrade for 2 hours. So essentially the entire point of this step is to dry the nitrocellulose membrane so that the DNA fragments that have been transferred onto it can now be affixed in their locations. So you can either achieve that by heating it in vacuum or in a regular oven, or by exposing it to ultraviolet radiation to permanently attach the transferred DNA to the membrane. Next, the membrane is exposed to the hybridization probe. I would like to add, before you expose the membrane to the hybridization probe, you have to block the membrane, because the probes are also nothing but DNA molecules. So any free region on the membrane where there is no DNA molecule already present can bind to the probe because of the ionic interactions between the negatively charged DNA molecule and the positively charged membrane. So in order to avoid that, you have to block the membrane, similar to how you block the membrane in Western blotting. So blocking has to be carried out using an inert solution. Okay. So the membrane is then exposed to a hybridization probe, which is a single DNA fragment with a specific sequence whose presence in the target DNA is to be determined. And the probe, mind you, has a sequence which is complementary to the sequence of the target DNA. The probe DNA is labeled so that it can be detected, usually by incorporating either radioactivity or tagging the molecule with a fluorescent or a chromogenic dye. So in some cases, the hybridization probe used can even be made out of RNA instead of DNA. Now, to ensure the specificity of the binding of the probe to the sample DNA, most common hybridization methods use salmon or herring sperm DNA for blocking of the membrane. So because it's a nitrocellulose membrane which is binding to DNA, so for blocking you have to use DNA. So commonly either salmon or herring sperm DNA are used. Okay. So they will block the membrane surface and target DNA. And sometimes detergents such as SDS is also used to reduce non-specific binding of the probe. So it can also be kept in the solution in which the probe is present, which is added to the blood for binding of, or to allow binding of the probe. Okay. So after hybridization, the excess probe is washed away from the membrane, typically using an SSC buffer, which is nothing but your saline sodium citrate buffer, which is commonly used in southern blotting. And the pattern of hybridization is visualized. So it can be visualized either on X-ray film by autoradiography in case of a radioactive or fluorescent probe, or by the development of color on the membrane if a chromogenic detection method is used. So if you're going for autoradiography based detection, your probe has to be radioactively labeled. If you're using a fluorescent probe, then you can detect it using some chemi detector system, which can eliminate the fluorophore at a specific wavelength and capture the emitted fluorescence. So this is an example of a southern blot visualized using chromogenic detection method. And this is an example of visualization of a southern blot using autoradiography. So to summarize very briefly the protocol that we talked about. So you start with the DNA. You digest the DNA to break it down into small fragments, and you run this digested DNA on an agarose gel to separate them on the basis of their size. For the purpose of reference, you have DNA markers. Next, you transfer these separated DNA fragments from the agarose gel onto the nitrocellulose membrane. So you have blotting papers on top, you have this transfer buffer at the bottom. So you have the gel placed in the transfer buffer. On top of that, you have the membrane, and on top of that, you have the blotting papers. So a suction force will be created by the capillary action, because water will try to move from here to the dry blotting papers, because the blotting papers will absorb the water, and as the water moves, it will also move the DNA fragments along with it from the gel onto the membrane. And now you have your DNA transferred onto the nitrocellulose membrane. So you first bake it, either you heat it in an oven or you expose it to UV light to fix the DNA fragments on the blot, and then you hybridize it with the radioactive nucleic acid probe. But before hybridization, do remember to block the blot. So once you hybridize, you expose it to the film and you get the autoradiogram. And or you can expose it, put it in the detector and collect the image from there. So this is a schematic representation of the transfer setup for southern blotting. So you have the sponge at the bottom placed within the tank, which contains your saline sodium citrate buffer. And then you have a filter paper, you have the gel, the membrane, some more filter papers, paper towels, and finally on top of it all, you have the weight. So the weight is placed to ensure application of even pressure on the entire system, and to ensure that there is uniform contact between the gel and the membrane, and that there are no air bubbles in between. And this sponge will begin to absorb the buffer from here, and because the paper towels that are placed on top are dry, so therefore the buffer will tend to move upward, and as it moves upward under capillary action, it will pass through here, and when passing through, it will take the DNA fragments along with it. So this is the transfer apparatus, what is typically used for southern blotting, and this is what it looks like. So a little bit about fish, very briefly. So fluorescence in situ hybridization, or FISH, is a laboratory technique used to detect and locate a specific DNA sequence on a chromosome. So essentially over here you're trying to detect a specific DNA on full chromosomes. So therefore, it is taking place in situ because you're not really, isolating the DNA and then digesting the DNA. You're just preparing a smear of chromosomes on a glass slide, staining it with your fluorescent label probe and visualizing it directly under fluorescence microscopy. So therefore, this visualization is in situ and hence it is called in situ hybridization. So in situ meaning where the DNA fragment originally exists. It is not being removed from its native environment. So in this technique, the full set of chromosomes from an individual are affixed to a glass slide and then exposed to a probe, which is nothing but a small piece of purified DNA tagged with a fluorescent dye. The fluorescently labeled probe finds and then binds to its matching sequence within the set of chromosomes. And now, using a special microscope, that is a fluorescence microscope, the chromosomal as well as subchromosomal location where the fluorescent probe is bound can now be visualized. So essentially, you have this chromosome, right? And however, you do have to keep in mind that you can't just directly add the probe to the chromosome. You have to treat the chromosomes affixed on the glass slide to allow slight denaturation so that your label or your probe can go and anneal with its complementary strand. So we are not really going to talk about how the sample is processed for performing fish because that is outside the purview of this lecture. I just wanted to give you an idea of what fish is. It's just a modified extension of Southern hybridization, and I just wanted you to be aware of what it means. So this is an example of how chromosomes are looking once they've been labeled with fluorescently tagged probes. So you see the chromosomes are counterstained using DAPI. So DAPI counterstaining is performed to help visualize where the chromosomes are. And within the chromosomes, wherever the probes have bound, you can see fluorescence. So here we are seeing, or we have the chromosomes have been labeled with probes or with two different probes, each fluorescing a different color. So we have the red one and the green one, and you can see that over here. So yeah, fish gives really beautiful pictures. So that brings us to the end of this lecture. So to briefly summarize, we talked about Southern hybridization, its origin and how it was named. We talked about what it is. We talked about the basic underlying principle. We briefly discussed upon how probes are designed for performing Southern hybridization, and then we went through a step-by-step protocol for the same. We also briefly discussed about fish or fluorescence in situ hybridization. So I hope this lecture was clear to you. In case you have any questions or any doubts, please feel free to leave it in the comment section and I will definitely get back to you. If you like this video, then do like and share it. And if you want to learn more about different techniques commonly used in labs, then do subscribe to this channel. And I'll see you again next time. Bye.